This leader is provided to permit the projectionist to complete adjustments before the film begins. The volume and tone are identical to the soundtrack which follows. This is a recent photo of a second-year medical student. It conveys a good deal of useful information. But it fails to tell us that she's a pretty girl. That fortunately takes 3 dimensions, which is the way we see, the way we comprehend, the way we think. In the same way, we need an instrument that can give us a comparable insight into the microscopic world. That briefly is the reason for the scanning electron microscope. Anna can easily distinguish the red blood cells by size and shape. But the image is only in 2 dimensions, as flat as a drawing on a sheet of paper. This same blood smear, in the scanning electron microscope, produces these unique images of red blood cells. The preparation of a blood smear for examination on the scanning electron microscope is fairly rapid and simple. First, the slide is put into a vacuum evaporator, where it is coated with a very thin layer of gold, no more than a couple of 100 angstroms thick. Biological materials conduct rather poorly. This treatment makes the surface electrically conductive, so it's ready to be scanned by the moving electron beam of the scanning electron microscope. The whole slide, not just a tiny fragment of it, is inserted into the airlock chamber. The position of the slide can be observed through an airtight port in the column of the instrument, so it can be checked and adjusted. The whole process generally takes about 2 minutes. Now we are ready to look at the sample of Anna's blood. On the face of a cathode ray tube, we see the result of this new method of imaging. A 3rd-dimensional view of Anna's blood smear. As with any microscope, what we see can be photographed and recorded. We feel we're in contact with the real thing because we see form as we are accustomed to see form, in the familiar parameters of size, shape, texture, and depth. If greater magnification is needed, one has only to turn a knob up to a limit. The form of the normal red blood cell is quite stable, and this biconcave pseudo-donut is probably much like the shape of the cell in the human bloodstream. The same is not quite true for more fragile specimens, like the eosinophil, where the original shape has been flattened out by drying in the air. But when the membrane shrinks, we can see the granules inside, as here from a patient with leukemia. We are only at the beginning in the present state of the art to connect the precise shape of the cell with the disease. This kind of red cell, the target cell, is often found in cases of thalassemia. Other bizarre cells are also found in patients with this blood disease. This form is characteristic of sickle cell anemia. Here are the strange shapes of red cells after cold agglutination in patients with acanthocytosis. Here is a whole human blood clot which has been fixed in formaldehyde for 48 hours. Now we have some idea at a glance of what a blood clot is really like. We can make out individual red cells enmeshed in bands of fibrin. Of course, we're still looking at the surface. How can we look inside? One possible way is to blast away the membrane with a beam of ions such as argon or oxygen. This cell has been etched in hydrogen for 15 minutes. This one for 35 minutes. The membrane has been blasted away so we can see what seems to be the structure underneath. The scanning electron microscope does not replace the conventional electron microscope or the light microscope, but rather offers an addition to our efforts to image biological systems. For example, a nerve tissue culture appears like this through the light microscope. However, by putting together a mosaic of from the scanning microscope, we can see whole sections of the culture in 3 dimensions. The neurons with their long processes, a kind of map in high relief which our minds can immediately grasp and understand. Precisely because we are walkers, touchers, seers in a 3-dimensional environment, the scanning electron microscope gives us Gullivers some insight into the Lilliputian universe. The eye is a window from which we view the passing world. Suppose we turn our instruments around and examine this marvelous organ. At first sight, it looks like an unknown planet. But we are looking at inner space, the epithelial cells of the human cornea. The outer cells are flat and transparent, and air drying has emphasized the junction of cell with cell. Here, for comparison, is a part of a rabbit cornea experimentally infected with herpes simplex virus. This egg-like mass is not the virus, but the infected deeper cells of the cornea now exposed. Deep in the ocular structure of the human is the trabecular meshwork, the complex filter through which the aqueous humor flows. Allowing for the effects of preparation, we can see the network of collagen fibers and the thin cells stretched between them. The familiar view of a diabetic retina through the ophthalmoscope reveals a great deal of information. The scanning electron microscope view gives additional information about the structure of the micro-aneurysm. And here is a living human eye, property of a non-diabetic second-year medical student. She's interested in the eye of the tribolium, the common flower beetle. This small creature can be examined in the scanning electron microscope while alive, because it has the unique ability to survive in a vacuum for about an hour. Like most insects, it has a compound eye. It also has an array of spines among the visual units. We can understand this arrangement at once, immediately, intuitively. What we can begin to grasp is the functioning of part with part, the connections, the architecture of living tissue. How does this new instrument work? Unlike the light microscope and the conventional electron microscope, The scanning electron microscope does not form an image by means of a lens. In the light microscope, light passes through a more or less transparent specimen and then through curved glass lenses in order to produce a magnified image of the specimen. It can be viewed directly by eye or can be recorded on a photographic plate. In the range where the light microscope produces useful resolution, the information content of the images is very good. Generally, the size, shape, and many architectural and chemical relationships can be determined. And of course, many living objects can be studied too. But the light microscope has a useful magnification of at most 1,500 times. In the conventional electron microscope, the image is formed by passing a beam of electrons through the specimen and then using magnetic fields as lenses to focus the electrons. The magnified image is visible on a fluorescent screen, or the electrons may be allowed to fall directly onto a photographic plate. Magnification of up to 200,000 times is common. Because the images produced by the conventional electron microscope are not within the mode in which we generally experience the world, a considerable amount of interpretation and technical skill is necessary in order to extract useful information from them. Here, for example, is a wire mesh, 200 square holes to the inch, photographed through the conventional electron microscope. It looks like no more than a flat shadow. But through the scanning electron microscope, it's clearly a piece of woven material. The quality of wovenness is apparent in the third-dimensional appearance which the scanning electron microscope can give us at once. Here is the jaw of a rotifer, as it is seen by the three methods of imaging. Essentially, the scanning electron microscope is composed of two systems: the probing system, into which the specimen is placed, and the imaging system, which displays the visual image. In the probing system, a hot filament generates a stream of electrons. The electrons pass through a hole in the anode as a high-velocity beam and are focused on the specimen as a point, about 1/1,000,000th of an inch in diameter. At the point where the beam hits, the electrons excite the material of the specimen to produce many kinds of radiation, including visible light, infrared, X-rays, and streams of electrons. A suitable detector is used to measure one particular kind of radiation leaving any one point on the specimen at any one instant. One of the most useful kinds of radiation is called secondary electrons. The measure of the number of secondary electrons leaving a point on the specimen is used to modulate a second synchronous beam of electrons, which is directed to the face of a cathode ray tube, resulting in a point of visible light. When the induced emission is high from a point on the specimen, the corresponding point on the cathode ray tube display is bright. When the beam of electrons is moved to another point on the specimen, radiation is induced from that point. This information can be displayed in a point adjacent to the first on the face of the cathode ray tube. The movement of both beams in synchrony is accomplished by a device called a scan generator. As the electron beam moves across each point on the specimen, corresponding points are visualized on the display screen of the cathode ray tube. In practice, the beam moves from point to point rapidly, and the series of points becomes an image of a line across the specimen. The specimen is scanned by the electron beam so that these lines follow one another rapidly. Any portion of the specimen scanned by the beam will be visualized on the cathode ray tube. The scanning electron microscope can magnify up to 20,000 times. To obtain magnification, the points and lines formed by the probing beam are packed closer to each other, so that only a portion of the specimen is scanned. Suppose we show you, as a kind of scientific game, some of the revelations of scanning electron microscopy and give you a couple of seconds to guess the answers. Can you identify this? No, it's not. It's the New York Times. Try this. No, it's not a cactus. It's a grain of pollen. Yes, this is the Spirochete of Syphilis, Treponema pallida. And this bizarre organism is a platelet, imaged from a smear of normal human blood. This looks like an archaeological dig. It's an ion-etched piece of tooth enamel, showing the various layers as they are excavated for study. This is part of the dental picture, too. It's a plaque scraped from the enamel wall. And the spheres are one kind of bacteria not yet identified, found in association with human teeth. And this is another, filamentous kind of bacteria from a carious lesion. E. coli, the normal guest of the human colon. Yes, this is the same organism, but after 90 minutes of exposure to penicillin, the cytoplasm escaped through the damaged cell wall. In the presence of higher concentrations of penicillin, a bulbous sac develops at the mid-portion of the wall. This outpouching is apparently the start of the development of an L form or spheroplast, a stage with a membrane but no wall. In this field, we see the full progression of the conversion of E. coli to L-form organisms in the presence of high concentrations of penicillin. First, the cells elongate. There are extrusions from the damaged wall. The formation of L forms. The membrane collapses. These are cancer cells from a breast cancer. The significance of this configuration is not yet known. But this is not pathological. It's neuronal tissue taken from a marine mollusk. The knobs are probably neural processes related to the synaptic junction. They have a topography very difficult to see by any other imaging method. Here are penicillin-resistant Staphylococci in the presence of penicillin. They appear normal. But in the presence of the antibiotic cephalothin, the bacteria swell and show granulations and protrusions of the cell wall. The full possibilities of the scanning electron microscope are still being explored. For example, this image of the flower beetle may be modulated by assigning a particular color to a particular intensity, thus getting a color image, not a color picture, since we are not dealing with visible light, but a kind of color diagram like staining, which greatly increases the amount of subjective information. Because finally we see not with instruments, not even with our eyes, but really with our brain, with our whole life experience. For the imaging of much biomedical material, the light microscope is still most useful. For extreme magnification, the conventional electron microscope is unsurpassed. It has brought us this picture of what may turn out to be the double helix of DNA. But for the marvelous individual geometry of life, as in this view of the rods and cones of the retina of a salamander, the scanning electron microscope remains a true and penetrating eye.